And we left off on this particular slide. I was just starting to acclimate you to where those pigments absorb, what part of the light spectrum, and this is the visible light spectrum, that those pigments, those chlorophyll pigments absorb the energy. So you have A absorbing the energy in one particular spot, and this is in the full length of the visible spectrum, and B absorbing the photo energy in another part of the spectrum, but not really that far off of it. And so pigments are substances that can absorb visible light. I've been saying this, but there it is in print as a term. The wavelengths that can't be absorbed are reflected or transmitted. And so this is the answer to the question that get asked, that you probably asked when you were in grade school somewhere, is leaves appear green because chlorophyll reflects and transmits the green light. So it reflects it, I guess the way we always explain it to children is reflects it back. So that's why we see the green. So you have chlorophyll A over here, and instead of having the numbers, you have the part of the spectrum that it's absorbing. Chlorophyll B, and then the only thing I've added to this one are the carotenoids. That will, again, remember, they are kind of absorbing a wider range, and they're the ones that absorb that extra stuff that keep that chloroplast from being injured. Plus all the light that gets absorbed is light that then can be used to drive the Calvin cycle. With regard to the design of this particular graphic, you have the capacity to absorb. That's on one axis, and then the wavelengths on the other. And what we are is we're given a particular wavelength, the capacity to absorb or the ability to absorb spikes. It also is absorbed at other wavelengths, don't get me wrong, except for, say, the lower, like 500 to 600. But the highest absorption happens at that particular peak number on all the three different pigments. The way we understand this, and this is a property of the various different pigments, is this is actually measured with a technique or a device called a spectrophotometer. And what this is a design that a scientist came up with. Measures the pigment's ability to absorb various wavelengths, and that actually you get a readout number. This machine, so the spectrophotometer, sends a light through a pigment and measures the fraction of the light that's transmitted at each wavelength. So how much of that light is transmitted? If it's not transmitted, it must have been absorbed. So this will give the readout the idea of what pigment is actually in there, what pigment is working to absorb that energy. Speaking of absorbing, you have the definition of the different ways to define this process, I guess we might say. The absorption spectrum is the portion of the wavelength that the light that the pigment is using. So the absorption spectrum is a graph plotting the pigment's light absorption versus the wavelength. So the absorption spectrum of chlorophyll suggests that ultra blue, violet blue, and red light work best for photosynthesis. So it's basically turning it into words, what part of the spectrum, as opposed to wavelength in numbers. The action spectrum which is the effectiveness of different wavelengths that are of radiation that are driving a process. So both of them are important. So the action spectrum is the spectrum most important for plant growth and metabolism, and the absorption spectrum is the spectrum of electromagnetic radiation that the light absorbs. So if we look at the graphic on the right, you have the absorption spectrum, and that looks pretty familiar. We've seen that before with the level of absorption between chlorophyll A, B, and the carotenoids. But this action spectrum mirrors it, but it's not quite as severe, because remember, that's a process. That's the rate that photosynthesis, the rate of photosynthesis. And this is measured by the release of oxygen, measured by how much of that byproduct of the Calvin cycle is given off. So this is the action spectrum is looking at how efficient this plant is actually using that light that was absorbed. And actually, the way this was figured out was the gentleman Endelman's experiment on bacteria, aerobic bacteria, with regard to what particular portion of the light spectrum that bacteria actually absorbed. In terms of a light, a pigment, when it absorbs light, it goes from a stable state or a ground state to an excited state, which is highly unstable. That instability then is used to drive a reaction. So when that excited or unstable electron falls back into the ground state, a photon is given off. And fluorescence is the effect that's created. So we have this fluorescence that's created because of this photon being given off. So you have a pigment that actually captures the energy. After it captures the energy, it is highly unstable, and that instability then is reversed, I guess you might say, when the electron then falls back into that stable state, but there is a product that is created that's called a photon. We can actually see it, the reaction visually in the fact that this illuminated this fluorescence that's happening. And you have a nice graphic here, photon coming into your chlorophyll molecule, exciting that electron, and then as that electron falls back down, you have heat and a photon given off. Photon in the form of fluorescence. Now into more of the nuts and bolts of how photosynthesis takes place. So the first thing we want to talk about is we're taking those thylakoids, we're looking inside in the inner membrane. What you have is a photosystem which consists of a reaction center complex, this reaction center complex. It's a type of protein, and that is surrounded by what's known as the light harvesting complex. So we have a protein. We're familiar with proteins. What we are is we're adding this reaction center complex and putting it in an area that is called a light harvesting complex. The light harvesting complex is the pigment molecule bound to that protein, that bound to it, so the transfer of the photons, so the light harvesting complexes transfer the energy of the photons to the reaction center. If we look at our image, we have a photon coming in, that's that yellow light, that yellow, I guess squiggly or almost arrow thing. That's hitting the green dot. The green dot is the chlorophyll. That chlorophyll then is moving that in what is actually the purple part, which is the light harvesting. Light harvesting is the portion that can grab the light from the environment. It then sends it down, it transfers it down. Remember, because if it grabs it, it's going to be very, very unstable. So it transfers the energy that eventually then gets transferred to a portion of the reaction center, which is where you find the chlorophyll A molecule. That's special chlorophyll. It's the protein molecule, or the pigment as we've called it, that can actually capture that energy, and uses the energy to move an electron to a higher level. So if we look at on the left again, chlorophyll absorbs light. That's step number one. That's the photon going into the green circle, absorbing light. Energy is transferred from pigment to pigment. The energy reaches the reaction center where chlorophyll A uses the energy to move an electron to its higher level. Then the primary electron acceptor captures that electron. You can think of the primary electron acceptor as being the NADP, capturing that and then moving it on to the next step. So this is all happening in the thylakoid space. You have a membrane there. The outside of the membrane is the stroma. That's stroma, and then the inside is the space. And then alls we've gotten this to is this primary electron acceptor in this particular step. And then after the primary electron acceptor, what we are looking here on the left, it looks very familiar. We've just added a few little pieces here. But the primary electron acceptor, which you can see that those terms on the left at the top of the purple block, that then is passing that electron down a complex of proteins that you can think of it as similar to basically similar to, and actually even called, an electron transport chain. And that then electron transport chain, which we're kind of familiar with, is going to be able to produce ATP. So let's go through this left one again in terms of energy of electrons. You have light coming in. That's step number one, being passed through these pigments, eventually gets to the pigment chlorophyll A, which here we're just calling it pigment 680. That then excites an electron up to the primary acceptor. That's the next step. There is a byproduct here. You're giving off, you also need to have oxygen or water come in. So light and then water comes in. So there's another little piece here, because the water is going to then give you the hydrogen ion. And then oxygen is given off at this step. So a little bit different piece here. So that step 3, where water is coming in, providing the electron, then oxygen is given off as a byproduct. That electron then gets excited, we know that part, and then down the electron transport chain to make ATP, and then given to photosystem 1. So one of the things that I left out, unfortunately, and I'll have to pick it up on the next one, is these are called photosystem 2 and photosystem 1, and I'll explain that on the next teaching time.